Thanks, Bob. Yeah, today I'd like to discuss some of the work that I've been doing in a place called Manus in Papua New Guinea, and and how this research is actually contributing to coral reef conservation and management in that area. And this is a partnership between the Nature Conservancy, the Center, and 3 local communities in Manus. So just to introduce the place, Manus Island is in that little yellow box up there, and it's the most remote and least developed province in Papua New Guinea. It has about 50,000 people. There aren't any roads, electricity, or plumbing. And the people who live in these communities have fairly limited income opportunities. The things that they can take out of the sea are things that they can dry, because there's no refrigeration or anything like that. So we're talking about sea cucumber, trochus, shark fin, and increasingly fishing for the live reef food fish trade, which is, of course, the capture of live fish that transport to Asian markets, and they fetch a fairly high price in markets like Hong Kong. Our study area is in the southeast corner of Manus, in that red box, and encompasses about 60 kilometers of coastline. So in this study area, we're working with 3 communities. Pere to the right, which has about 1,000 people, Locha with 60 people, and Tawi with about 200 people. Now these communities consist of the Teton people, and the interesting sort of cultural aspects about the Tetons is that they're entirely reliant on marine resources. They don't own any land, so all the resources that they harvest themselves are taken from the sea, and then they trade with inland communities for agricultural products such as sago and things like that. The inland agricultural communities don't have any direct access to the sea, and that's just sort of a cultural delineation between the groups. As is common throughout Melanesia and in other parts of that region, the management of resources has sort of devolved to the local level. So within these community tenure areas defined by these orange lines, within those areas, each community has the authority to enact regulations and enforce those regulations, and those regulations are in turn recognized at both the provincial and national government levels. So in Bob's talk, he talked about local action, and this is really the scale, at least in this system, at which you can actually get management happening. You have to work with the communities because they own these resources. Now, one of the key resources for each of these 3 communities is a multi-species group or spawning aggregation, indicated by these red triangles. And we're actually working on one of the species that aggregate at these spawning sites, and this is the square-tailed coral trout. They aggregate monthly throughout the year, but they have a peak spawning season, and during that peak spawning season in Manus, you get about 1,200 to 2,000 individuals aggregating over a very small area. The adult spawning size ranges from 31 to 56 centimeters, so these are perfect size fish for the live reef fish food trade. They sit very nicely on a plate. The IUCN recently classified this species as vulnerable, and that's largely due to it being a major target of the live reef food fish trade. Now, in 2004, the Nature Conservancy was approached by some representatives from the community, and the community members were basically asking for some assistance with understanding these aggregations, because, as I said, they're a key resource for the community. So a monitoring program was initiated in 2005 where the Nature Conservancy trained community members in scuba and actually monitoring techniques, and so the aggregations monitored monthly. And these trend lines are for the 3 aggregation sites. You've got 2005 to 2010 along the x-axis, and the peak spawning density, number of fish per 1,000 square meters. Now, the trends for Pere are in red, Locha in yellow, and Tawi in blue. In 2005, the live reef fish trade came in, a Malaysian company came into Manus and went to each of the communities and asked for permission to fish. Pere and Locha said, no, we're not interested. Tawi actually allowed fishing to occur, and over 6 months, that company removed 6.5 tons of coral trout. And for those efforts, the community was paid $100 per community member. So they were pretty happy with that deal. But as you can see, from 2005 to 2006, we had a 70% reduction in the peak density in the aggregation. And that aggregation hasn't recovered much, and in fact has declined. Overall, there's been a general decline in all 3 aggregations, and that reflects a lot of internal fishing pressure, and also, according to the community members, the increasing incidence of nighttime spearfishing. These coral trout are really susceptible to nighttime spearfishing because they rest at about 3 to 5 meters at night, and they're pretty easy to shoot. So with the availability of underwater torches, these guys are going out at night and taking quite a few. Now in 2009, one community, Pere, enacted some management measures, and I'll talk more about those later. And what we saw in that aggregation between 2009 and 2010 was a 60% increase in density, whereas the other two aggregations continued to decline or remain stable. Now, when the initial few years of monitoring were reported back to the communities, this generated some concern, obviously, from the communities, and they said, look, we really want to figure out what's going on with these aggregations and what we can do to protect them. But there's a few key questions that we have that we want help answering. The two main key questions, the first one is, if we protect these aggregations, are we going to get any benefits on a local level? So they're not interested in closing off their aggregation and then exporting all the benefits to adjacent communities. And that's really been the focus of my ecological research, is trying to understand connectivity and larval dispersal. And so I was contacted through the scientists at TNC to ask if there was something that we could do to try and ask this question. So where do the adults go when they leave the aggregation site? And where do the larvae go that are produced by that aggregation? And that sort of gets at that question of whether local action is going to result in local benefits. The other question that they had was, is there anything else we can do or any other habitat that we can protect to facilitate the conservation of these aggregations? And one of the key issues there is, where are these things recruiting to? What's the recruitment habitat of juvenile coral trout? So at that point, we initiated this research partnership between the Nature Conservancy, the Center, and these three communities to try and address these issues. The methods that we used, we worked closely with each of the communities, and we used local fishermen to sample the adults at the aggregation sites. So on these little small sites, we're capturing the adults, we're tagging the adults to try and understand where they're going after they leave the aggregation site through tag returns from fishermen. And we also took a genetic sample from each adult. We then employed local spearfishers to search for and sample the juveniles, and this was actually probably the toughest part of the field work, because the local fishers had no clue where the recruits were. They're too small to be captured in any kind of line fishery, and the spearfishers don't target small fish. So they probably just had never really paid attention much to where these little recruits were. So we ended up spending several weeks and employing about 50 to 70 spearfishers to search virtually every reef in that 60-kilometer coastline area to try and figure out where these things were. And unfortunately, there was no published information about where they went. When we finally did find where these recruits were going to, we took a genetic sample again, which we were going to use then to compare to the adult genetic sample in a parentage analysis to try and understand where the dispersal was happening. We also used an otolith analysis to understand juvenile growth rates to give us some idea of the population dynamics of the species, and try and understand how those fishing pressures might be impacting the growth and conservation of the population. So in 2008, we sampled over 4 months and collected 1,000 adult samples and 500 juveniles. In 2010, in May this year, we went back and sampled 450 adults in 1 month, and I'll be going back there again in a few weeks to sample the juveniles. So we'll have 2 years of information. Now, because of the lack of infrastructure and facilities in Manus, we had to get a little creative in doing this sort of field work, and so this is basically our home and research platform. And we would just park these things about 7 or 8 kilometers offshore. We had local guys out there helping us all the time, and this is what happens to a canoe when you let a bunch of boys hang out on it for a few weeks. So all the cooking and sleeping and sample processing is all done on that canoe. We don't touch shore for about 3 weeks, and the platform that we're living on is about 11 foot by 7 foot, and we had about 9 guys living on there. So we thought inviting any of our female colleagues would probably be a bit too much to ask, so although they probably would have improved the condition of our work site. And the way this worked is the local fishers would go out. These are guys sampling the adults at the aggregation site. They paddled their dugout canoes from their villages and spend all day out there catching fish for us, bring the fish back for sample processing. We take the adults and we measure them, we sex them, we give them a tag, we take a fin clip, and then we release the freshly sampled fish back to the aggregation site. And then for the juveniles, we're sending spearfishers out all over that study area and collecting juveniles, bringing them back. And here we are at night pulling otoliths by torchlight, which is very cozy. And you can see these little juveniles aren't going to be wasted. They're already on plates ready to go. So nothing's wasted. So what are the results from this work so far? The question about where adults go. Here we've got the spawning site in the red triangle for locha, and the green diamonds actually show where the tagged fish have been captured off the aggregation site. They're not moving very far, a few kilometers away in most cases, and a lot of them are staying within the boundary of the community. Some do cross boundaries, that's to be expected, and not very problematic. The recruitment sites for this species are highly localized and very discrete. I mean, as I said, we searched all these reefs across this whole area, which was a big task. And the recruits are concentrated in particular areas. And what you'll notice is there are recruitment sites in each of the 3 community tenure areas, and they tend to be directly behind the aggregation sites or in close proximity to the aggregation sites. This makes us think from a connectivity and larval dispersal standpoint that there might be, in fact, a lot of larval retention from one aggregation into that same general area, although unfortunately the parentage analysis that will answer that question definitively is still about a month away from completion, so I don't have much to say on that issue yet. But we're hopeful that, and Jeff will be talking about some of the work that we're doing in the Keppel Islands, we're hopeful that we'll see similar things here and that local action will actually benefit the local communities. The Mana sits at about 2 degrees above the equator, and the growth rates of the juveniles are pretty extraordinary compared to a lot of the coral trout that you see in the southern Great Barrier Reef, for example. Within about a year, just over a year, they're reaching spawning size. So a fish can be born in May of one year, and by August the next year, it's on the spawning site, which is pretty remarkable. So these things are growing like gangbusters. I think they must have a lot of food. What I'd like to focus on now is that this partnership between the Nature Conservancy and the Center and these communities has really had a lot of sort of flow-on benefits for conservation and management as a result of the interaction between all 3 parties. We're feeding in scientific information to them. They're facilitating that scientific work by telling us where to go and how to fish and actually doing a lot of the work themselves, which is great. So it's given us an opportunity over the years to really interact with them and discuss management options and what can and can't be done and what should and shouldn't be done. In the largest community of Pere, where the overfishing is probably the most acute because of the large population size, we actually worked with them for the last couple of years in drafting a management plan, which was signed into legislation in 2009. And this management plan essentially puts fishing restrictions on about 35% of their reefs. So this is a large community that depends entirely on fishing. Got a little weird, but depends entirely on fishing, and so this is a significant investment really in the future of their reefs and their fisheries. So they established 3 marine protected areas with a variety of fishing restrictions. There's no take of trochus or sea cucumber or corals allowed within those MPAs. They've banned all kinds of gear, including spear guns, nets, scuba, poison root, which is pretty widely used throughout Melanesia, dynamite, crowbars, things like that. They do allow periodic harvest by community consent, but that requires all the people in the community getting together and saying, yes, we need to harvest this for a festivity or something like that. There's also one cultural area. Again, all the gear bans in the MPAs are also replicated in the cultural areas. And again, harvesting is allowed for community festivals. This is Pere's community tenure area, and the green boxes or outlines define the MPAs. These MPAs include the spawning site and also some of the juvenile recruitment habitat for the coral trout. The blue area is the cultural area, and that really protects essentially what is the historical location of the Pere community. It also protects some reef flat habitat, which is an important breeding ground for some of the sea cucumber. Now, one of the nice flow and benefits of having this management plan and getting it signed into legislation and having it recognized by the provincial and national governments is that it facilitated the community to go and get a grant from the National Fisheries Authority of Papua New Guinea. And they used this grant to basically come up with an alternative livelihood that revolved around fishing. And that was the installation of a fish aggregating device about 1 kilometer offshore of the outer reef. Now, we went back in May to just have a look at this fish aggregating device and how it's working. And what was remarkable is between 2007, when there was no fish aggregating device, and 2009, or 2010 rather, I can tell you that there was a massive decrease in the number of canoes fishing on the reefs. They were all fishing this aggregation, or this aggregating device. And so this is now the primary location for the community to fish, and the primary location from which it receives its food, its fish both to eat and to sell on to the agricultural communities and trade for surplus stuff. In fact, we were in the markets in the main town in Lorengau, and they said, hey, you guys did a really great thing by getting that aggregating device out there, because now we've got plenty of fish to sell. And they all had little tuna out there and little dolphin fish to sell. So it's a very simple device, but very effective. This is an underwater shot looking at a little school of tuna up in the top there, cruising by, that will probably soon be on someone's plate. Another great flow-on effect, and Bob touched on this a bit, is that we now have Locha and Tawi saying they want to do the same thing. They want us to help them generate a management plan that achieves similar goals to Perry's management plan. And we also have communities that we haven't been working with that are just hearing by word of mouth how these things are working out. They now want to get involved. And in fact, the Nature Conservancy has just received a large grant from AusAid, which we hope to tap into to design an island-wide network of MPAs for Manus. So I guess the key point is that this research partnership between the Nature Conservancy, the Center, and these communities is actually succeeding in answering some key scientific questions, but it's also been really beneficial for catalyzing local action. And I think that it's starting to result in some great benefits for reef conservation and fisheries management, and hopefully we're going to see those benefits spread throughout Manus and beyond. I'd like to just finish by thanking my collaborators here, especially the communities of Perry, Locha, and Tawi for all their hard work and for their permission to work with them. Rick Hamilton at TNC, some of you may know him, he's been a great inspiration for me, and just has great connections with the community and a great scientific background. This is really helping me to sort of bridge that gap. There's a number of key collaborators, and I'd like to also just finally acknowledge funding sources from US-based organizations, the Nature Conservancy and the National Fish and Wildlife Foundation, and also the generous support of the ARC Center of Excellence.